as you have seen how a 10 nanometer fiber is folded into a 30 nanometer fiber to make the higher level of folding or organization. But still the this level of organization or this packaging of 10 nanometer fiber into 30 nanometer fiber is still insufficient to fit the nucleosomes or the chromatin fibers into a chromosome or into a nucleus. So there needs some additional folding to compact the DNA further. The exact nature of this higher order folding is still unclear. However, some popular model proposes that the 30 nanometer fiber forms loops of about 40 to 90 kb long. So these 30 nanometer fibers, they are further folded or condensed, and they form loops, and each loop is about 40 to 90 kilobase pair long. These loops are held together at their bases by a proteinaceous structure which is referred to as nuclear scaffold. So as you might be remembering, in case of prokaryotes, the protein core was present, and around that protein core the supercoiled loops and some relaxed loops were present. Similarly, here in case of eukaryotes, there is a nuclear scaffold in the center. It makes the core, and around this nuclear scaffold the 30 nanometer fiber is present, and it makes loops like this. So in this way higher order folding is achieved. The true nature of this nuclear scaffold, it remains mysterious. So let me summarize whole condensation process. So you can see this is a DNA molecule, and the first level of folding or condensation is the formation of nucleosomes. So in this at this level, the DNA molecule wraps around the histone molecules, and after this wrapping or winding, it forms a fiber. This fiber is called 10 nanometer fiber, and which resembles the beads on a string. And this the further condensation or further level of organization is achieved when this 10 nanometer fiber, it makes 30 nanometer fiber. And you know, this is achieved either by zigzag model or by solenoid model. So this is a 30 nanometer fiber. And the further level of organization, in the further level of organization, this 30 nanometer fiber, it makes loops around a nuclear scaffold. So here this is a nuclear scaffold, and around this scaffold these loops are present. So this is the next level of organization, and then this scaffold, it makes the chromosome. So in this way, starting from this DNA molecule, there are several folds of organization, several levels of organizations to acquire a chromosome.